Alright, so back again, and this is the third video, hopefully the last in this series before we finish up topic 2. And what we're going to be talking about now is an example where selection pressure over evolutionary scales, over multiple generations, has been able to drive the evolution of protein structure, actually changing the protein structure, in this case of an ion channel, and a predator that feeds on a very toxic prey item. And I have a couple of videos here to introduce this topic. First, I'll just show this one, and then we'll show the longer one that explains it in more detail.
This innocent-looking newt enters the frog's field of attack. The frog strikes and swallows. But the battle's not over. The newt's skin contains a deadly poison. Within minutes, the frog is dead. And it's the lowly newt who struts away victorious.
Alright, let's switch and watch this one.
Student 37 years ago, my professor told me a story about 3 hunters out here in the Coast Range being found dead, and there was a newt boiled in their coffee pot. Nice one. So his question to me was, go find out if these newts are poisonous.
Edmund Brody Jr. has studied the rough-skinned newt ever since, the last decade or so with his son, Edmund Brody III.
This is probably the most poisonous animal in the world, with enough skin toxin to kill tens of thousands of mice or perhaps 100 people. Of course, they don't bite, so this isn't really dangerous holding it unless I were to eat it, which I won't. The question was, why should a small animal like this be so many times more toxic than necessary to kill all predators?
The Brodys discovered another animal is responsible.
I'm gonna head over to these brambles.
Okay.
The common garter snake thrives in these parts. Harmless to humans, it feeds on earthworms, frogs, and toads. But there's one prey in the snake's diet a few other predators ever touch.
She's got a food item.
Oh, yeah.
Look at that. It's a pretty big object. I don't know if it's big enough to be a newt, but it could be. Come on, honey. Here it comes.
Yeah, here it is.
Hey, how about that?
It had eaten a full-sized male newt.
Yeah. This species of garter snake is the predator that we think is driving the evolution of the high toxin levels in the newts. This is the only thing that can survive an encounter with a newt. It's the only thing that can, therefore, represent a selective pressure for increasing toxicity. As the snakes get better at resisting the effects of the toxin, the prey has to evolve higher levels of toxin. You can think of this as this sort of escalating, counter-escalating arms race between the predator and the newt, the prey.
But the toxin does take a toll. Some snakes are slowed down. Others are immobilized for a few hours after eating a newt. In the lab, the Brodys can measure the garter snake's resistance to the toxin. They coax a baby snake down a track wired with motion sensors and record its time.
Time?
3.4.
Then they inject the snake with a small amount of purified toxin to simulate the effects of eating a newt. Now the snake is raced again. A snake with low resistance can be stalled to a standstill. A resistant snake is much less affected by the toxin, but it too pays a price. The more resistant a snake, the more slowly it moves without any toxin.
The snake experiences a cost from evolving the resistance. That snake would be more susceptible to its own predators. So there's a trade-off between speed in a snake and the level of resistance.
It's now abundantly clear that evolution is driven not just by physical forces, such as storms and fire and climatic change, but much more by biological forces. That is, particularly the way species interact with one another, cooperating with one another, parasitizing one another, preying on one another.
What made the lion fierce and the zebra fast? What sparked the development of tooth and claw? The deadly dance of predator and prey drives evolution.
All right, so pretty interesting evolutionary story here where you've got this predator-prey relationship, this very poisonous newt here, which makes tetrodotoxin. It's actually one of the most poisonous compounds, man-made or in nature. And remarkably, these snakes have evolved the ability to eat them. And so what we know is that this tetrodotoxin has a very negative effect on a critical ion channel that regulates muscle contraction. So the snake requires this ion channel in order to move, in order to breathe, to move its lungs. And so this toxin here, if you were to be exposed to it, you would basically cause respiratory arrest and you would stop breathing. But the snakes need to eat, and the snakes want to eat these poisonous newts, and so what they've done is they've evolved their ion channels to become less sensitive to the toxin. Not completely insensitive, but at least less sensitive. So here's the structure of the ion channel here, and it has many transmembrane domains, and there's critical amino acids that are all colored, which are modified in these, in only the groups of snakes. This is an evolutionary tree, many different species, including Homo sapiens on the bottom. These guys are all the snakes. The rest of these are all the snakes. And what you're showing you is that all of these in bold with the newts by them, these are the ones that eat these poisonous newts. And they each have evolved slightly different ways, different amino acids, to allow them to eat these newts. All of which share the common property of reducing the ability of the toxin to affect the ion channel. So just an interesting case of coevolution between making this toxin and modifying an ion channel in order to be resistant to that toxin. But there's also many other examples of the evolution of proteins either matching the environment or matching some predator-prey relationship. All right, so now we've gone through several examples of how enzymes, in some cases substrates, can be changed on an order of very rapidly, such as almost instantaneously, to the order of several generations. And so these are 5 examples shown down here. And what I want you to do, your task is now to put these in order from fastest to slowest. All right, so go ahead and try this on your own before take a break from the video. Try this on your own on your Top Hat account, and then come back and after you're done answering the question. Okay, so I'll help you out with the solution here, and basically what you have is a series, and you're trying to first, let's just start off with trying to find what might be the fastest. Okay, so let's see, we start increase cell size. Oh, that's kind of slow. That's happening on the order of days to weeks to months. Secretion of a hormone, that's pretty fast. That might be a possibility. See, change in primary structure, that's evolution, so that's definitely not the fastest. Let's see here, phosphorylation of protein. Yeah, that's really fast, on the order of milliseconds, almost instantaneously. Upregulation of protein, that's minutes to hours to days. All right, so let's go with 4. 4 is the first one. So that's either these two. Can rule these two out, and then what's next? Well, they both have 2 next. Yes, secretion of, so that's the fastest. Second would be secretion of hormone, right? So that takes us through here. That doesn't help us though. Answer the question, and then we have 5 or 1. 5 would be upregulation of protein versus increased cell size. Well, I'm probably going to go with upregulation of protein transcription translation, minutes to hours. All right, so that would be 5. Next would be, let's see, increased cell size. Yep, that makes sense. And then finally, changes in a protein's primary structure, which would be evolution. So we would go with this one as our answer. All right, good job. All right, so this brings us to the last learning objective for topic 2, in which we're basically going to look at receptors. Remember, receptors usually serve as the sensors for the negative feedback systems within the cell, and we'll talk about where they're found within the cell. We'll talk about how they mediate a signal, what happens after they're activated downstream from them, and examples of the signals or ligands that they respond to. All right, so again, remember, this is our cell circuit, negative feedback, and we're talking about the sensors. These are receptor molecules, and later we'll talk about signal transduction as well. All right, so first receptor we need to talk about are nuclear receptors, and these reside in the nucleus. It's shown right here, intracellular nuclear receptor. And what happens is these bind usually to, these are a very simple cascade, just usually one protein. And what happens is the ligand is usually lipid soluble. They can go through the lipid bilayer, go through the nuclear membrane, and bind directly to the protein, the nuclear receptor, and activate it, and allowing the transcription of genes. Gene mRNA. All right, so it's a very simple type of signaling. They usually bind to lipid-soluble ligands, which can diffuse through the membrane. And so this is a case of a receptor. It's not often that a receptor doesn't have to be on the surface of the cell. And the reason why this works is because these ligands can go through the membrane. They don't have to bind to the membrane. They can diffuse right through it. All right, so these bind to things like steroids. So your anabolic steroids that control muscle growth, your sex steroids that control your testes, testosterone, estrogen work this way. And there's also a lot of toxins, unfortunately, in the environment that can mimic this. So here's estradiol or estrogen, and you can see that there's a lot of compounds that are produced. A lot of these are human, man-made contaminants, things that come from like pesticides like DDT, things that are made due to plastics, and these can unfortunately mimic these signaling molecules in your body and cause all sorts of problems with your signaling cascades. So these are examples of nuclear hormone receptors, and there's about 50 or 60 of these in your genome that respond to various lipid-derived signals. Okay. All right. So for the vast majority of others, though, the ligand is usually water-soluble and cannot simply diffuse through the lipid bilayer, and so there has to be a receptor here in the membrane. And these are binding to extracellular chemicals that are water soluble. So the idea here is you have an extracellular messenger or ligand here, and that will bind to the outside of the receptor in the membrane. And this, in some cases, this can allow something like this class are ligand-gated ion channels, so they allow an ion to move into the cell or out, and that then triggers a series of events that leads to the cellular response. And so one example of these are acetylcholine-gated sodium channels. We'll learn a lot about these when we talk about muscles, because this is how your muscles are triggered to contract, your skeletal muscle. So acetylcholine is the ligand, and that's released by neurons. It binds to this receptor and allows sodium and potassium to flow in and out, and that initiates muscle contraction. It's also present in your central nervous system and controls nerve conduction as well. And there's actually got quite a few of these. There's quite a diversity of these, over 100 of these, in terms of generally of ligand-gated, not just acetylcholine, ligand-gated ion channels. Very important for neurotransmission and neuromuscular transmission. And these bring us back to the very introductory video that I showed you of these snails that harpoon fish. And that's exactly how at least some of their toxins work. Here, normally acetylcholine is released from at the neuromuscular junction, binds to the acetylcholine receptor and allows muscle contraction. However, if you've been stuck by one of these snails, you've got this purple guy lying around which actually plugs these holes and doesn't allow the acetylcholine to enter. And because of that, you get a paralyzed muscle, and the fish can't breathe or move. And in your case, you would have respiratory arrest because your diaphragm and your muscles around your lungs would not be able to function. All right, so there's many other examples of ion channels that don't bind to ligands, but they're actually activated by physical stimuli in the environment. And you have some that are sensitive to voltage. You have some that are sensitive to touch, pushing of the skin, touch receptors all over your body. You've got some that are sensitive to cold and to heat. And so all of these are ion channels that don't respond to a specific ligand, but actually respond to physical conditions, such as temperature, voltage, or pressure. All right, so another very important group of receptors are called G-guanine, G-protein coupled receptors. And these so-called G-protein coupled receptors have a series to initiate a signal where you have the G-protein receptor, they bind to a G-protein, and then finally an enzyme is activated. One of the most common scenarios is you get a ligand binding here that activates the G-protein, which activates the enzyme. In this case, the enzyme produces a second messenger called cyclic AMP, and the cyclic AMP then initiates a signal within the cell. Right? So it's a handoff. You're handing the baton off to these two stages. Eventually you get an enzyme that's activated, which then brings about some sort of downstream effect. All right, so these are very, very prominent. There's over 1,000 of these in the genome. These are used to sense many odors and smell. These are also used to detect light in your eye, and also allow you to detect adrenaline in many of your different tissue organs. So this again, odors allows you to detect light, and it's actually how adrenaline is signaling throughout your body when you have excitement, such as jumping out of a plane. Then you have a smaller, another group that are called enzyme-linked receptors. These are more simple because it's just usually one protein, and what happens is the ligand binds here, and when it does, it activates a second messenger. Right? And so you have these. These are important for detecting things like insulin, growth factors that regulate tissue development, and also things like atrial natriuretic peptide that regulates urine formation and the amount of sodium in your urine. Okay, so that takes us to the last learning objective, is where we're going to be talking about the second messengers. These are the things that function usually downstream from the initial receptor to push the cascade on further. And we'll talk about why these are called second messengers. And then we'll talk about some advantages of why you have so many signaling molecules, as well as some disadvantages. All right, so the idea here is we've already tackled this, the receptors. Now we need to tackle this. And this is where you have second messengers and protein signaling. And what these tend to do is transmit the signal throughout the rest of the cell. The receptor usually cannot leave the membrane, so it has to hand off the signal to other proteins or small molecules, which can then diffuse through the middle of the cell and carry the signal throughout. And remember that essentially almost all signaling within your body is due to physical interactions, one chemical or protein interacting with another. And so this is a series of physical connections. These molecules have to literally bump into each other within the cell for this to happen. Right? There is no network of electrical or audio carried signals within the cell. It's all small molecules, proteins binding to each other, right, in a series. And so here's an example of this. We have, in this case, it's a G protein coupled receptor, activates a G protein, which activates an enzyme. here, a protein, and that turns on a second messenger, makes a second messenger, so that the idea is that the first messenger is this ligand outside the cell, and the second messenger is the one inside the cell, which is now transmitting the signal inside instead of outside of the cell. It then activates things like protein kinases, which then activate a final effector enzyme and bring about the cellular response, and that's your negative feedback. All right, so this looks just like this. We have our sensor up here, your receptor, you have signal integration down here, your second messenger and protein kinases, then you have your effector and your response. Now note that in many cases you have an amplification of the signal, right? You have a massive amplification or multiplication of the signal. So you may have a single extracellular ligand here. Let's imagine, say, adrenaline, a single adrenaline molecule in your blood. You're excited, you're scared. It gets released, it binds to this receptor, activates, and that can activate multiple G proteins, which can then activate multiple adenylyl cyclases, which can then activate multiple cyclic AMPs, which can activate multiple protein kinases. And then finally a second enzyme is activated. And so there's usually many steps, and these steps are often multiplied. And so what are a couple of advantages of this? Well, I've already mentioned one. You've got amplification, and the other is integration. Because oftentimes how strongly you respond to your first messenger will depend on other events within the cell, other things going on. And so you can, by having so many different levels, you can have other inputs from other pathways. Let's say that you are responding to adrenaline, but in some cases you might want to moderate exactly how much you respond to that, depending on what time of day it is, what kind of diet you have. And so you need to modulate exactly how strongly you respond. All right. So one output of adrenaline is to release glycogen from your liver, and under different levels, under the exact same level of adrenaline, you may not always want to produce the same amount of glucose released. Glucose released from the liver. You may want to modulate that depending on exactly how much glycogen is stored in your liver, how much glucose is already in the blood. And so in order to do this, you have to have other inputs to this, other ways of interacting. And so usually this isn't a simple, just straight cascade. It's usually a network of signals, and you have other inputs into this to modulate exactly your output, in this case, how much glucose gets released. All right, so there's your two advantages. All right, so then think about possible disadvantages. What might be a disadvantages of having so many different components here, so many different things interacting? All right, and basically it's just anytime you have increased complexity, you have the increased problem of something going wrong. All right, and one example of this is cancer, where you have regulation of the cell cycle of mitosis, and this is a very complex cascade of many different protein kinases and other proteins functioning together to influence exactly when and how often your cells divide. And things can go wrong. The more things that you have controlling a system, then any single one of them going wrong, eventually you get a few of those going wrong, and you can have the cell cycle running out of control. And that's how you have tumors form. Right? So complexity allows you to very fine-tune regulate things, but it also can lead to problems when things start going wrong. All right, this is just an example of the amplification or magnification of the signal. So a single epinephrine molecule binding to a liver cell can actually release over 1 million glucose molecules. And the way that works is you may have one single ligand, extracellular ligand or first messenger. You can have 10 adenylyl cyclases activated, and then each of those may activate 100 different cyclic AMP molecules, which then can activate in this case, we're now at 1,000 different protein kinases, which can each activate 100 different enzymes. You get 100,000 enzymes activated. Each of those can catalyze production of 100 glucose molecules. So in the end, you have 10 million glucose molecules from one epinephrine or adrenaline molecule. All All right, so this is just, lastly, this is just a little summary of the different types of second messengers. So one common one is going to be cyclic AMP, which is basically just a cyclic version of a nucleotide. You take ADP and you modify it. Another is cyclic GMP, where you start with GTP. This often leads to regulation of protein kinases. Another one you can have is nitric oxide. This is actually a gas that's produced from an enzyme. Nitric oxide, among other things, regulates the diameter of blood vessels and the dilation of blood vessels. In one place this is important. Well, I don't know how important it is, but certainly it's something to be aware of, is actually in the male penis. This regulates erection. And so drugs like the blue pill, the name escaping me right now, are the ones that the way they work is actually to increase nitric oxide to cause dilation of blood vessels in the male penis. So you have to affect erectile dysfunction. All right, so another important one is calcium. Calcium, of course, is an ion, but it's also a small molecule signaling. Normally the calcium concentration in the cell is extremely low, but it is high in the endoplasmic reticulum and outside the cell. And so you can have things like a G protein activating an enzyme called diacylglycerol, which will lead to cleavage of some phospholipids, and you get this molecule called inositol triphosphate, which is a piece of a phospholipid which can go and activate channels in the ER to allow calcium outside into the cell. And then once calcium is released into the cell, it can mediate all sorts of signaling. All right, so that's it for the slides. I do have a few top hats for you to practice, and so what I think I'll do is I'll post these for you and have you guys work on these on your own as homework, and then in class then we can go over these together. Okay, so I'll end the video now. All right, thank you very much, and again I apologize for not being able to make it live to the lecture today.